All right, this video should finish up this chapter and get us ready then to look at specific areas in the nervous system. So here again, this whole chapter 12 is all really devoted to how does nervous tissue work. So we've looked on the cellular level. Now we want to kind of expand a little bit when we talk about graded potentials, is this idea of, well, we don't always get a depolarization and action potential generated and one cell talking to another cell, right? We have other options for what can happen between these neurons. And we'll move then into neuronal pools to talk about, well, now we have to consider a lot of different neurons talking to each other at once. So let's start here with this idea of graded potentials. So we were looking at this idea again, local changes in the membrane potential starting with the dendrites, right? And if the local change was high enough, right, we reach a threshold, then we would see an action potential generated that would move down the axon, right? And then move to the next neuron in line, okay? So again, this change correlates with the strength of the stimulus, right? So how much, and the change we're talking about is really the idea, this change is related to how many sodium channels opened. So the more sodium channels open, the more sodium enters, the more depolarization we get. We hit threshold and we are going to change, basically. So I say here, right, two choices. If sodium or calcium enter the cell, right, because both of these are positively charged ions that are high outside of the neurons. If either one enters the cell will depolarize. It will become less negative. As you see here, we're going from resting membrane becoming less negative. Here, 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 and here. But you notice in this scenario, only this rate change is enough to generate an action potential. Right? So if sodium or calcium enter, right, depolarization occurs, and they are higher outside and they're positive, and we consider this excitatory, right? If potassium leaves, right, so if potassium channel opens instead, or chloride enters, and chloride is negatively charged and it's high outside the cell, then the inside becomes even more negative, and we refer to this as hyperpolarization that you see happening here, even more negative, and these are inhibitory. So this nice figure again, small stimulus causes small depolarization, doesn't make threshold, no action potential. Larger stimulus, right, causes more, but still not enough, right? A stimulus of longer causes a longer lasting depolarization, but again, the strength. It's about the strength. Here we have a larger stimulus that depolarizes above threshold, above that, right, negative 55, and this may trigger, or should trigger, an action potential in the following, the neuron, the next neuron in line after it moves down the axon of this neuron. All right, some stimuli result in hyperpolarization, which, again, if it's potassium or chlorides, you hyperpolarize. So for me, what you need to keep straight is which ions lead to which events. Okay? All right, summation. So now we're going to look at a figure like this to really get this idea of summation. So adding everything up together, all these changes. So here in this cartoon, we have a number of different, right, neurons converging on this one neuron, its cell body and its dendrites. Right here we see it as it's a cartoon still, but again showing it more with the more of the axons coming down here. So this cell body, or this cell body, needs to electrically determine how much of a change has occurred and if that is enough to generate an action potential. So here they didn't identify this here, but on this one they identified 1, 2, 3, 4 of these as excitatory. That's what the E stands for, right? Postsynaptic or presynaptic potentials, and 2 inhibitory, right? So excuse me, postsynaptic potentials or inhibitory. So you excite or you inhibit, right? So that is in reference to whatever these neurons are that are upstream or up here, right, and are now going to affect this neuron. So postsynaptic potentials, again, because here we're talking about postsynaptic is this is the postsynaptic. And the potential, what change has occurred? They're formed in the receiving neurons. this one or this one, and they get excitatory and inhibitory, some of each. Again, in this cartoon, we've got a few more excitatory than we have inhibitory. So we see, again, if they're exciting, we see this summing, more of a change here. But then we factor in the inhibitory, less of a change, right? And there's no action potential. More stimulatory, some inhibitory, and then finally that fourth, right, excitatory one takes us to threshold. So, but if it's inhibitory, it's hyperpolarized, or it's right, really causing repolarization. If it's excitatory, it's causing depolarization. But what this neuron does, right, this receiving neuron, depends on all of these added together, the negatives and the positives, negative, negative, positive, positive, positive. So if I hit threshold, then, right, and this process of summation, we will see this cell body generate an action potential, an excitatory one, that will move down its axon. So I say here again, hypothetically, what is a cell to do? It has to listen to all these, right, other cells that are talking to it. So a cell body may be receiving both, as we see here, and the sum of the excitatory and inhibitory will decide if an impulse is generated or not. All right. So synapses, again, we've been looking at this. These are our communication links. So once it moves down, so this one is coming into this receiving one, and then this one is sending on, possibly sending on. So every time we have a joining of these, basically the axonal bulbs with the dendrites or the cell body, right, we have this situation, right, where the axonal terminus, right, has all these vesicles, and these vesicles contain what we call neurotransmitters. And we looked at this in the last chapter on muscles. And the neurotransmitter there we focused on was, of course, acetylcholine. Now, in that situation, the acetylcholine had a very short distance to go, and it diffused across, and these were, again, ligand-gated, because ligand is a chemical, right? And ligand-gated channels with these receptors for the neurotransmitters, right? And again, if there were no receptors here in the postsynaptic neuron, so this area right here, then nothing would happen. But with these receptors for the neurotransmitter and these receptors associated with channels, we're going to see some change occur. Is it depolarization or is it hyperpolarization? So I say here, electrical, right, ions are passed from one cell to the next. If one cell is depolarized, it will depolarize the next. Right? That's a relationship. The chemical, so we talk about electrical chemical communication, is the neurotransmitter. All right? All chemical synapses, so this is, again, we're going from electrical change to a chemical change, and then again to electrical change once we bind. So all chemical synapses have, right, a presynaptic part, a neurotransmitter, synaptic cleft, right, so the space between the two, receptor proteins, here they are, postsynaptic part, and neurotransmitter eliminated or taken back up. So in the muscle chapter, we eliminated, right, we had this enzyme, acetylcholinesterase, that destroyed the acetylcholine, so we could stop the response as soon as we needed to. So I say this is really the same situation as we had when we had the motor neuron communicating with the sarcolemma and the muscle cell. All right. So transmission of the stimuli, right, or action potential. So this action potential, right, walking you through, arrives at the presynaptic, right. So here's the bulb, the axonal bulb, causes, right. So here's cause and effect, causes voltage-gated calcium channels to open. So we're adding this. So we're backing up a little bit, and we're saying, okay, action potential comes to the end and stimulates this change, right? Voltage gated, to change in electricity, opens these gates. Calcium moves into the cell, and it's calcium entrance which causes the release of the neurotransmitter. And again, we've only talked about acetylcholine, but there are quite a few other neurotransmitters. The neurotransmitter is released from its vesicles, exocytosis, right? It moves, right? So it diffuses across the space, right? Binds to a specific receptor on the postsynaptic membrane, which is shown here in blue and then highlighted here or zoomed in here, causing channels for sodium, potassium, or chlorides to open or close. Okay, so if these open, right, the sodium ones, we get depolarization. If potassium or chloride open, we get hyperpolarization. So which channel opens in the postsynaptic depends on the neurotransmitter released and the types of receptors in the membrane. All right? So the neurotransmitter systems, all right, here, just to let you know, this is actually not something I will test you on, but there are about 40 different ones that we are aware of, and there's probably more. But some neurons produce only one kind, and others can have 3 or more different kinds of neurotransmitters. So there's cholinergic systems, and this was the acetylcholine ones, the ones we talked about. And they do have different types of receptors, nicotinic and muscarinic. And just for you to know, there are amino acids classes. That's a class of neurotransmitter: glutamates, GABA, glycine. Biogenic amines. These are made from amino acids, like serotonin comes from tryptophan, dopamine, epinephrine, norepinephrine from tyrosine. Right? There are neuropeptides, which are short chains of amino acids, like the endorphins. Right? All of these things are made in the rough endoplasmic reticulum of the cell body, which is why you see all that, the Nissls bodies, or what we call the chromatophilic substance. And they are transported down the axons of the nerve, of the axon, transported down the axons and stored in vesicles at the axonal terminal right bulb. All right? The effect is actually pretty complicated, so we're really not going to expect this, but this is, again, we're introducing these concepts. So ultimately the effect depends on, again, the neurotransmitter released and the receptors. All right, so when acetylcholine binds a nicotinic receptor, the postsynapse is depolarized. If it is a muscarinic, it might be hyperpolarized. So again, it could be the same neurotransmitter, but the receptor is different in the postsynaptic membrane, and that changes what happens. So again, this would be inhibitory, this would be stimulatory. Glutamate is excitatory since it causes depolarization, while glycine and GABA cause hyperpolarization. So biogenics, instead of changing electrical potential, may affect gene transcription or some other metabolic event. It's pretty, again, we're really not expecting you to understand this, and your book does not give you all the, a lot of detail on this either. So just to let you know, I mean, it's the nervous system is complex in this idea of summation and receptors and neurotransmitters. All right. What limits this response? And I mentioned this a couple slides back. Some is regulated by enzymes, like we talked about in the muscle, right? The acetylcholinesterase. And these enzymes are found in the cleft and on the postsynaptic membrane, and they degrade the neurotransmitter, right? They get rid of it so we can stop the response. So we have a quick way to start and stop the responses, what we are doing. There's also reuptake, where the neurotransmitter is transported back, right? So endocytosed to the presynaptic membrane or even nearby neuroglia cells. And then there's also the possibility for destruction or removal. The destruction or removal of this neurotransmitter allows for the control, the fine control we have between nerve cell to nerve cell or neuron to neuron, and the ability to re-stimulate as is needed. Okay? I said, got to throw this in right as we get here to the end. In case you didn't already feel a little overwhelmed by your nervous system's complexity, we do mention in the lesson, this again is not for testing, a couple classes of receptors, the ionotropic and the metabotropic, and I say, these are ligand gated, these are complexes of membrane proteins that affect chemical processes, so they themselves can be involved, and they're showing you the two examples here. Binding to the ionotropic here and the metabotropic here. The neurotransmitter is considered a first messenger, binds its receptor. A G protein is activated that influences another protein, right? This effector is an enzyme which produces a new molecule, and so forth. This is a cascade event. We are not going to need to know this, but this is why we talk about the nervous system as having some complexity that is certainly not in this understanding of an action potential, but is definitely in the understanding, in the application of what happens.